I'm going to go over some vocabulary about how we discuss the DNA strands in transcription that may be more challenging to understand from reading. So imagine we have a piece of duplex DNA. We know that DNA runs, it's double-stranded and it runs anti-parallel. So I'm just going to show you where the ends are. But we're actually going to have a transcription bubble, an opening between the double helix where transcription takes place. And I'll draw RNA polymerase, that's the enzyme responsible for synthesizing RNA in transcription. So just like DNA polymerase, RNA polymerase only synthesizes nucleotides in the 5' to 3' direction. And so I'm just going to label the bases. This separated strand here is, this is called the transcription bubble. And I'm going to label bases in the transcription bubble. For example, G, C, T, A, C, ... Okay, so we have our double-stranded DNA. This is the strand that is the template strand. Notice how transcription goes from this side to over here. And the template strand is going to run, has to be the 3' to 5' because synthesis is going to be anti-parallel and it can only go 5' to 3'. So this strand here is the template strand. Our sequence of messenger RNA is going to be complementary to that. Because RNA polymerase is going to base pair with these bases as it synthesizes. That's what it uses as the template. The template strand is called the non-coding strand. And I said it's going to be complementary. And because of that, it's called also antisense. And another way of designating this is the negative, the minus strand. This strand up here, is called the coding strand. It runs in the same direction as the messenger RNA that's coming out. See, 5' end over here, 3' end over here. Our synthesis is going from 5' to 3'. The sequence of the coding strand is the same as the messenger RNA. With one exception, and that is that messenger RNA does not, or any RNA does not have T's. T's will instead be replaced with uracils, U. This strand is referred to as the plus strand because it's the same as the messenger RNA, and it's also called the sense strand. So our sequence of messenger RNA, if we were to start transcription here, our messenger RNA would be complementary to these bases. So that would be the base that's complementary to A is T, but in messenger RNA it would have to be U, T to A, G to C, C to G, G to C, T to A to U, A to G, dot, 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 3' direction. And you'll see if I label the bases in that coding strand, they're going to be the same as my messenger. This is my messenger RNA here. This would be T, A, C, G, C, A, T, G. So quite a bit of vocabulary, and hopefully a bit of an orientation in how we describe the DNA strands in transcription.